So today we are going to take a database question of IB biology higher level. This question is basically the topic genetics. You need this combination of transcription, translation, and genetics. So let's look at this question. It's saying that GPR 120 is a transmembrane protein that functions as a receptor for a long chain unsaturated fatty acids. When fatty acids outside the cell binds to the receptor, so this is a receptor. When these fatty acids are present, they bind to the receptor change shape. As a result, signal is passed to the interior of the cell. This causes an increase in the intracellular calcium concentration, which has a wide range of effect on cell activity. So basic information you must have that in a cell, then gene expression, we are talking about here gene expression, and they are the receptors. So here this is GPR 120 is the receptor, which is for long chain fatty acids. Long chain fatty acids can come attached to this receptor, and it initiate the expression of gene which cause the increase in the calcium ion concentration. This is what we understand from the question so far. Now a strain of mice was developed that did not produce 120 GPR protein. Now these group of mice were fed for the age of 5 weeks until they are 16 weeks. All right, on a high fat diet. So there's a high fat diet and there's a normal diet provided to the mice who do not produce GPR20. So that indicates that if they do not have GPR20, they don't have the receptor, and then we look at what happened to their body mass. Now there's a control group as well. So you can see here that this one is in a normal diet. This is high fat diet. GPR deficient, this protein receptor is not present. In this case, receptor is also not present, but only the difference is in the diet. Now the question is asking you to compare and contrast. So here we have to tell in similarities and difference both. The body mass of GPR 120 deficient mice and the control mice. So we have to talk about the compare and contrast between these two and these two and these two. According to the question, you have to talk about three things. Compare and contrast the body mass of 120 deficient mice and the control mice on a normal diet, and then the high fat diet. So we see that the normal diet, in the normal diet, GPR deficit is same as the control, while in the high fat diet, GPR deficit is more than the control. All right, how about the normal diet and high fat diet? The mean body mass of normal diet is less than high fat diet. So mean body mass of high fat diet is higher than the normal diet in both cases, right? So 3 points we talk about there, and I'm going to show you the mark scheme. What 3 points you can write down there precisely is, it's A. No difference on normal diet between control and GPR. So we talk about that. No difference in normal diet between control and GPR. Both high on high fat diet than a normal diet. So GPR also high, control also high. Then GPR 120 deficit higher than control on high fed diet. GPR deficit, this we talk about, is higher than control on fed diet. So when you're answering these type of question, you can plan beforehand before you phrase the question here. Now next question, next information they have given, the gene of GPR120 protein is located on chromosome number 10. It is a autosome, right? Not a sex chromosome. The allele for GPR120 from 312 extremely obese adult. So sample size you have 312 and children were base sequenced. So base sequence were taken. 6 alleles were discovered that differ by one base from the wild type allele. So the allele which is like the common wild type allele, only 6 were different in one base sequence. They are likely to have been produced by a base substitution mutation. 2 of the mutations cause a change in amino acid sequence of the GPR120, which is missense mutation, but the other 4 synonymous mutations. So let's talk about these terminology which I highlighted. So first of all, autosome 10, chromosome 10 is an autosome. All right. Then you see that sample size is 112. Then you see base substitution mutation. What is base substitution mutation? Is a change in a single base. So here you need to have a theoretical information that a codon is made up of, we call it triplet codon, ATC for example, CGA for example, AAT. So if there's one base change, a single change caused in the base, so for example A, this become C and C, CGA, AAT. So this become a base substitution mutation. Now when there is a 2 of mutation cause a change in the amino acid sequence. So what they are saying that not all mutation, there are 6 mutations, 6 alleles were discovered, so 6 mutation happen, but not all mutation cause the change in the sequence, only 2 mutation cause amino acid sequence change. Now here you should know the information, theory information I'm talking about, that you have bases, and based on these triplet codon your amino acids are arranged. What happened? So you know this is triplet codon, amino acid 1, 2, 3, right? But if this is amino acid A, B and C. Now what they are saying, because there's 6 change happen, so this change happened, but this is still A, B and C. This change happened here, but this is still A, B and C. But when this change happened here, this no more remain A, this became D, B and C, something like that. So only 2 happen in this way. So 2 terminologies are there, missense mutation and synonymous mutation. Synonymous means it is the it remains the same, though there's a mutation happen, but the sequence of amino acid will remain the same in the synonymous mutation. But if missense mutation happen, means now there is a change. This is something like this, a change happen. Now let's look at the question. Explain how only some of the base substitution mutation cause a change in the amino acid sequence of the gene for GPR120. So why only 2 cause the change while this remains the same? The reason is degenerate more than one. As I told you, base substitution change a codon. Amino acids are coded for different codons. Several codons can code for the same amino acid. So how many codons we have? We have 64 codons. Just for make you understand, I'm telling you this information. So 64 codons we have, and we have not 22 amino acid. We have, sorry, not 64 amino acid. We have only 22 amino acid in our body, while we have 64 codons. That means that these codons, there are more than one codon coding for the same amino acid. So I for example, if this change happened, but still this codon is still actually coding for the same amino acid, so it becomes the synonymous. So that's why we are saying several codon can code for same amino acid. Several codon can code for same amino acid. Base substitution change a codon. Amino acid are coded for by different codons. So B and C almost same. Phrasing is slightly different. And A. So this is 2-mark question. You can write down A and B or A and C. All right. Now look at next question. Using the position of chromosome 10, identify which two mutations were closest together in the gene. So we go back, we look at which two. So this all 9 is fine. Same, same, same. Now change start here. So we'll see 1, 1. These two are 1, 1, 6, 6. 1, 6, 6, 6, 6. 1, 6. This is changed, this is changed. So there are three changes here. Let's look at the 3, 3, 1, 3, 7, 0, 3, 1, 3, 7, 0, 1, 4. So there are 2 change. So we put here 2. This one 2, 5, 8, 4, 6, 2, 5. Oh, this is very long difference, 4. So the closest is the one which is having the least difference. So the least difference one is 9, 5, 3, 3, 7, 0, 3, 1. So 9, 5, 3, 3, 7, 0, 3, 1. And the second one is 9, 5, 3, 3, 7, 0, 1, 4. 9, 5, 3, 3, 7, 0, 1, 4. So these are the closest together. All right. The frequency of 2 alleles that cause a change in amino acid sequence were measured in 6942 unrelated obese humans and 7654 control. This is a sample size. The results are shown here. So you can see that the obese allele frequency. What is allele frequency? The frequency of that particular allele. So for example, when we are talking about here, the receptor protein is forming. So let's say this is for capital R, capital R, capital R, small r, small r, small r. These are allele. So frequency of capital R is this much, right? In these. So out of 4, this is 3 is capital R out of 4. Frequency of this particular allele. This particular allele, if I'm looking at these two, is 1 over 4. Or if I'm looking all 4, so it depends that what is the allele frequency. So after getting the frequency of allele, we found this is 5.5. Obese, control is 4.3, 2.4, 1.3. Outline the reason for using large number of obese humans and control in this research. Why earlier we had only 312 something, but this time in thousands we are taking sample. Sample size gives you a reliability of data. Reliability of data comes from large sample size. I'll show you the mark scheme as well, but first we understand. Reliability comes when there's a large sample size, so always is a standard answer. But in this case, especially the obese, there's a variety of reasons that can lead to obesity in humans. So because of the variation in genetic proposition for obesity, large sample size must be taken. Understand? So I'll show you. These are symbol 1. All other things increase reliability. We can identify anomalies, which is completely. So I talk about increase reliability. More genetic variation you will see in obese people, and to run some statistical test you need to have large sample size. Some allele frequency are very low, so you can see up there, you can talk about that, and anomalous result you could have spoken about. The association of one of the allele with obesity was statistically significant. So what we see statistically significant, then the other, you can see, you can take the percentage The difference here, I think it's if you count, it should come somewhere 9.94% or something. It is 97. Which one is higher? The difference is higher, so that should be your because the larger percentage difference between obese and control. So larger the percentage difference, that you should identify as the more statistical difference. Okay. Now, a clone of human cell that combined the wild type allele was genetically modified by inserting either another wild type allele, WT, or one of two mutant alleles. So, what we look at above scenario, two mutant allele, one is this, one is this. And we are looking at the intracellular concentration. What happened to the concentration of calcium when linoleic acid concentration we are changing, which is a long chain unsaturated fatty acid. So you can connect the previous information that this is an unsaturated fatty acid, this is a receptor. We know when this unsaturated fatty acid attach, it causes the concentration of calcium ions to increase. Now when I'm changing this, I'm changing the mutation happened to this one. So what happened to the concentration? Increase or decrease? What exactly happened? So we will look at there. Now we see that when it is the we are taking the clone and genetically modified with the another wild type. So is the wild type this one. You can see this is causing increase in the calcium concentration, while the alleles, these two, they are not high till the concentration is 10. When we increase the concentration, then only they increase. And the third one is it increase but not as exponentially as this one. Okay, so I draw, but this is better I remove. You can see the line clearly. All right, now what is they asking? Suggest advantages of genetically modified a clone of human cell for use in this experiment rather than using cell from obese people. So whenever you are doing a clone, that the first advantage of these thing is to controlling the variable. You can control the variable more appropriately. So you can see that first advantage is control variable. Now again, because it's a lot of difference, we talk about obese people, the variation could be a lot. Cells from obese people will have lot of differences. We do not know because of which difference exactly we are getting that particular result. So that's why control variable, the only difference will be genes. So here, because we can do the repetitions, so only difference we can do, only difference here we can control, only difference is type of genes. So you can take wild type, or you can take mutated type 1, or you can take mutated, and it is very easy to repeat experiment. All right, now look at the next question. Outline the effect of, so this is, you can I just now when I was explaining you the graph, I outlined the effect of linoleic acid concentration. So when it is the wild type, let me double check what they're asking. Only with the wild type we are talking about. So you can say that increase with the concentration increase, and in the graph above, first it increase, then the increase is smaller. So it's exponentially increasing until here, then it becomes smaller. All right, so that you can explain here. And then the compare and contrast of two mutant alleles. So compare and contrast two mutant, I explained you already in the graph. Let's look at the mark scheme. Both allele reduce calcium concentration with the same amount at low. So both, when they both are present, they reduce. It's less than the normal one. So both reduce when they are at low concentration, but with higher concentration, R67C increase exponentially and reach almost to the level of the wild type, while R270H remains low. So still low with high with R7OH, but as high as WT with R67. All right, so we explained the graph. Now, last part, biologists have often debated whether characteristics in humans are due to genes or environment or combination of both. Here, its question is discussion. So you can discuss with whatever information has provided, but you have to give the evidences. So I will explain, then we will look at the mark scheme. So first part, when we see the graph in this question given, that there is a change, it's not, this is also deficit, only thing we are changing the diet, right? High fat diet and normal diet, and we see diet is external factor, right? It's not genetic proposition. So when we look at the diet, high fat diet, given the body mass increase, it indicates that external factor is affecting the body mass. But when this is first evidence, when we talk about this one, this is not giving you this one, you can talk about obese human, the percentage change, it is genetic. You can see how the percentage change in allele frequency. So here we are talking about the genetic proposition evidences. This one also you can talk about genetic proposition evidences, because we are changing the, we are using the mutant gene. So any point you can discuss along with the evidences. So let me show you the answers. Argument for both factor, mice on high diet had higher body mass of deficit mice was higher than control, so this become genetic. High fat diet will give, then you can explain with the linoleic acid concentration graph. So both factors are affecting with this. Diet having more effect because you see more increase in mass with the high diet. Gene not being the only factor, because there are difference in allele frequency. We can see the difference in allele frequency, but some non-obese people have the same allele as obese people. But this information is actually not very appropriate, because from the question you are not getting enough of this information. You can't provide the evidence for this one. So it's better, always better to give the information which you can provide with the evidences from the question. So that's all for your data response question of genetics, and good luck for your IB biology. I'll come back.